gonna switch to English. Thank you. So my talk will deal a little bit about immunity and recombination, not exactly in that order. This is the classical. This is nothing now. It's the classical picture that we present for representing the cause of the biological infection and immunity. And as you probably know, when the animal is infected, very rapidly develops a viremic phase that can be of different duration related to the age of the animal, to the strain that we have in this particular case. But sooner or later, this viremia disappears. And normally it's something like this. You have a peak about day 7, 10 of infection and then fades out for after some weeks. And what we have is that after this viremic phase, the virus persists for a period in the body. And normally we can find the virus in the lymphoid tissue and finally disappear. The tonsil is the place where we can find for a longer period normally the virus. And this is a special case because if we think of what we call normally a persistent infection is an infection that persists for the life of the animal or for the life of the person. And that's normally because the immune system is unable to control that. This is not exactly the case in PRRS. In PRRS, the infection is finally cleared. So that means that the immune system is doing the work, but needs time to do that work. The immune system is able to clear the infection, but needs time to do that. And if we look at this immune response, what we normally have is a very early development of antibodies. When you do an experimental infection, about 50% of the animals are positive in the ELISA after 7-10 days in the cause of the infection. And by day 21, all animals are normally positive if they were naive when infected. The interesting point here is that these antibodies that we have in the early response are not having neutralizing capacities. They are not able to clear the infection because they cannot neutralize the virus. If we look at the neutralizing antibodies, normally they appear after 3 to 4 or 5 weeks after the onset of the infection. And as we will see later, sometimes we have antibodies that are in a very low amount, in a very low titer. Some other animals produce a little bit more, but normally this response is very limited in the initial infection. When we look at the cell-mediated immunity and interferon gamma, the measuring of the frequencies of interferon gamma secreting cells specific for PRRS is the normal parameter that is examined here. What we see something like this, ups and downs for several weeks, and finally, coincidentally with the development of the high titers or the higher titers of neutralizing antibodies and with the clearance of the viremia or the infection in the body, these responses start to increase. This is not what we expect from a common infection, acute infection. If you look, for example, in influenza, what you could see could be like a compression of everything. Everything will happen very rapidly in influenza, and that explains why we have a very short period of infection. This is the classical viremia that we have. For about 4 weeks, 5 weeks, we have a viremia, then disappear, type 1, and similarly for type 2 in 2 different experiments. Let's talk a little bit about the adaptive immunity. We know that the neutralizing antibodies have the capability to block the infection, and this is an experiment done some years ago in which the authors transfer, by passive transfer, homologous neutralizing antibody to piglets, and they challenge the animals. And as you can see here, and as we can see here, animals that were transferred titers of about 1 to 8 of these homologous neutralizing antibodies were protected or were mostly protected against the development of the viremia by day 7 after challenge. Anyway, some of these animals were infected and the virus could be found in lymphoid tissue, so this protection was not 100% but was very substantial. And this was a proof that these neutralizing antibodies can protect. But what happens? This is what we call a heat map. A heat map is made by It looks really complicated, but it's really easy. A heat map is something like this thing. You produce hyperimmune sera against a panel of viruses, again a panel of strains, and then you check the reactivity everybody against everybody, how the antibodies raised against this strain are able to neutralize this other strain. So the darker the red, the better the reactivity. The lighter the color, the lower the reactivity. If you look here, we can identify, just let's follow the picture. You can identify that for some strains, reactivity with other strains is very high. And for some other strains, reactivity with other strains is very low. So indeed here we have a problem. It seems that some strains are able to induce antibodies that have more pan-neutralizing capacity compared to the others. But this is not that simple, because when you look at the animals, also you have animals that produce or seem to produce specific antibodies for the strain that infected the animal, and some other animals, probably after repeated contact with the virus with different strains, probably after maturation of the immune response against the virus, are able to produce what we call broadly neutralizing antibodies. So the picture is very complicated. There is a site that is on the virus. Is this virus, this strain, inducing more or less broadly reactive antibodies, and the other site is in the repertoire of the antibodies that have a given animal. When we look at the cell-mediated immunity, the picture is similar to that. In this experiment, we analyzed the response using this ELISpot method that is measuring the frequency of interferon gamma secreting cells against the homologous or the heterologous response against the homologous or the heterologous response using the same two strains. Experiment number 1, animals were infected with A, and then we tested against B. Experiment number 2, animals were infected with B, and then we tested against A. So we crossed the things. And what we could see here is that in this case, homologous was better than heterologous in all cases. In this case, was the opposite. So it's not that simple like saying homologous is always better. It depends on the strain, and it depends again on the animal. So this is the global picture of the immunity. Very simple, but I think that you have a general overview of that. Let's talk a little bit about evolution of the virus. The first thing that we have to understand is that evolution is a natural fact. All living creatures, maybe viruses are not living creatures, but we can accept this figure. All living things evolve. And if we look at the evolution, one of the driving forces of evolution is mutation. Mutation is very powerful to create new things. And looking at the mutation rates that we have for different organisms, you would see here, we are here higher eukaryotes. Our mutation rate is in this range, 10 to the minus 11. So that means that if we take the global picture of all the living things, our mutation rate is very low. But our genome is very big. If we look at RNA viruses here, the mutation rate for RNA viruses is very high, in the range of 10 to the 3, 10 to the 4. But the genome of these viruses is relatively small. If you think of PRRS virus, round numbers, the PRRS virus genome is 15,000 bases. So that means that with a mutation rate of between 10 to the 3 and 10 to the 4, probably all copies of the virus that are produced in the body carry some mutation. Okay? Why are these mutations produced in RNA viruses? Because the RNA polymerases are hard workers. They are very good at producing copies, but they are not very accurate, and they lack the ability to correct the errors that they may introduce during the replication. So in other words, we are producing copies of the virus, and once in a while there is an error introduced in a place in the genome. Instead of a T, we put a C. Instead of a A, we put a T, or something like that. Okay? So what happened with these mutations. If we consider this mutation rate of 10 to the minus 3 minus 4 nucleotides, that means that every copy of the virus has a mutation. And the result is that we have this virus in the body present in the form of a quasi-species. A quasi-species means that we have infinite or almost infinite variants for this virus in the body. The results of these mutations can be very different. Let's imagine that the mutation produced a change in an amino acid in one of the viral proteins that is essential for the virus. And by changing this amino acid, this protein lost the function that is corresponding to that. So we have a non-functional protein, and of course, this variant will disappear from the population as a non-viable mutation. Or maybe we have a mutation that results in a neutral change. Since the genetic code is redundant, maybe the mutation is encoding for the same amino acid. Or maybe we are changing an amino acid that is not changing the function of the protein because it's in a place that is not very relevant for the function of that given protein. These variants will eventually fade out. When you look at this, it's like they appear, they disappear, they appear, they disappear, but there is no special gain on having that mutation. Or maybe that mutation may result in a beneficial change. And in that case, that variant may or will prevail. What type are the changes that are beneficial? This can be because this particular variant is better adapted to host, or is better for replication, or maybe because you are changing something in an epitope that is critical for immunity. So, we know that we have this constant mutation. We know that not all mutations will accumulate in the genome. But if we put this in the context of the big figures, one infected animal will have millions of copies of that virus, and we will have probably millions of animals that are infected. So in practical terms, that means that these changes will accumulate over time in a given lineage of the virus. The second driving force that we have for this type of viruses is recombination. And recombination is something that is natural. Recombination is one of the mechanisms by which RNA viruses evolve. How this recombination happens? For recombination to occur, we have a number of things that should happen. The first is that one cell is infected with two different viruses of the same species. And normally it's said that recombination is more likely as closer are the two viruses that are found in the cell. And the second thing that we have to consider is this recombination rate. The recombination frequency is estimated for coronaviruses that are in the same order than PRRS, taxonomically are very close to PRRS, is estimated in the order of 10 to the minus 4 or 10 to the minus 5 per site and generation. So that means that every 10 to the 4 or 10 to the 5 copies that you have in the case of this co-infection can have what is called a breakpoint, a recombination point. Again, this seems to be a very low frequency, but if you consider that you have millions of animals infected and some of them will be co-infected by two different viruses, this is not that unlikely. How this recombination happens in this type of RNA viruses? Very simple. You have one polymerase. Remember, this RNA polymerase is very hard-working, but is not very accurate. You have two different viruses, very closely related one to the other, so the final result is that one or more of the copies of the genome copies will have a part of a strain number 1 and a part of a strain number 2. And this is different from what we are told for influenza. The phenomenon that we see in influenza is completely different. When we talk about the diversity in influenza viruses, is because mainly of mutation, but again with reassortment. And this is because influenza has a segmented genome. Different pieces, different genes in different segments that can go together in very different ways. Completely different mechanism. Let's take a look to the molecular mechanism of this recombination. The most common mechanism by which recombination happens in this type of viruses like PRRS is what we call template switching. This is our genome of the virus, this is the polymerase, and the polymerase starts to replicate this genome, okay? Since this polymerase is really promiscuous, sometimes what we see, or what can happen, is that the polymerase, as he says here, disassociate from the template. You have the polymerase, the template, you start producing a copy, and by chance they separate, okay? If there is another template in the cell, maybe the polymerase can start replicating this other template. And what can happen? The polymerase stick to a non-homologous template, for example, a messenger RNA in the cell. The result is something that is non-viable, so this will not go anyway, this will be destroyed in the cell. Or maybe you have a homologous template, something that is very similar, another PRRS strain, but that is not exactly the same. What could happen if we have this homologous template? One possibility is that replication continue in a different position. We have to produce a continuous genome from nucleotide number 1 to nucleotide 15,000, okay? So if we stop in nucleotide, I don't know, 1045, we have to continue 1046. But maybe instead of this position, the copy starts in 1100. So there is a jump forward or a jump backwards. When this happens, the result is an insertion or a deletion. You are inserting an extra piece or you are cutting a piece in your genome because you jump forward or you jump backwards. The result of this normally is a non-viable thing, but depending on the place of the genome that this happens, we can have something that is viable. For example, in PRRS, if we compare PRRS1 with PRRS2, a big segment of a protein that is called NSP2, non-structural protein 2, can have a lot of deletion. So that means that many parts of this non-structural protein are very flexible and may allow this recombination. In other cases, you have an exactly match with the previous thing. So you finish your first replication in 1045 and you stick to the new template in 1046. So that's continuously perfect. You have a recombinant. Or you can go back to the original template and again you can create insertion or deletion or whatever you have in the middle. So one first question that we have for understanding this recombination is whether or not we have hotspots for recombination. Do we have some points in the genome that are more likely to suffer this recombination or not? There are some places where I can cut and paste more easily than in the others. It looks like that, but we are not 100% sure. And the next question is how we can detect this recombination. And detecting recombination can be a little bit tricky because for real detection of recombination, normally we have to combine at least two different methods. How we detect recombination? The most straightforward approach is to use an algorithm, a bioinformatic approach, analyzing the sequences that we have. And we know that with the software that we have in hand right now, just by using one of the algorithms, sometimes we produce some non-very accurate predictions. So normally what we do is to have two different methods. When we have positive results in two methods, it's pretty clear. I will show you some pictures later. And we combine that with the phylogenetic analysis. So one first thing, to be sure that there is a recombination, you have to apply a special analysis and you need to screen and to confirm. Second, as I told you some minutes ago, recombination may occur in different parts of the genome, and that means that may occur in OR5, this has been proven, but also may occur in different parts of the genome. So examining OR5, that is the usual gene that we sequence, is not enough to detect recombination. If you see it, it's okay, but if you don't see it, this doesn't mean that there is no recombination in that virus strain. So full genome sequence is recommended for analyzing recombination. And the third important point, once we detect recombination, what we could say. Detection of recombination does not allow to do any prediction in general. Why? Because virulence or attenuation is multigenic. So just by seeing that we have a recombinant, we cannot perform an accurate prediction of the characteristics of that given strain. Okay? How recombination looks like. This is a phylogenetic analysis of a number of samples using OR5 in this case. Okay? If we examine this OR5 with these programs that I'm telling you, the program told us that there was a breakpoint in position 219. And the meaning of this breakpoint is something like, if you look from 1 to 219, this set of sequences show one type of clustering. If you look at them after 220 until the end of OR5, the clustering is different. And you can see here this very clearly. This red strain here was of the green type in this first segment of the gene, but was of the blue type in this other segment of the gene. And this is what we expect exactly from a recombinant strain. We have one part that comes from one parental, another part that comes from another parental, and we can follow that by using this strategy. How frequent or it's important, this recombination, it's frequent, can we find it? Okay, I'll show you some results about that. These are the results of the examination of different strains, European type of strains, and the way in which we represented data is very simple. If you have one strain in which all segments cluster together, we put the same color. If we have segments that cluster with other strains different than the one that we are examining, we add another color. Look, for example, to this one. There is a portion of this strain that comes from the black strain, a portion of this strain that is red, a portion that is blue, a portion that is yellow, a portion that is gray. That is what we call a mosaic. This strain is the result of frequent recombinations in the field, and the same can be taught for other strains that we have in this picture. So recombination is common. How common? That's very difficult to say. That's very difficult to say because if you look at this is OR5 is here. By looking at OR5, we only have a very partial picture of that. In my guess, after our results, probably 5%, 7%, and proportion like that of the circulating strains are recombinant. Recombinant with other wild-type viruses, recombinant with other live viruses that we are using for vaccination. And the other point was how the virus can evade the immune response and how this relates maybe with this fact of recombination. I show you this picture that I think it's pretty interesting. This was produced in a paper that was aimed to understand how resilience to pairs can be controlled by the host. The idea was to identify genes in the host that can control infection. Okay, I'm not interested on that at this point, but for doing that, these people follow hundreds of animals during the course of infection. And for some of the animals, can you see this rebound in the viremia? So most of the animals have the nice curve of the infection. They were viremic. They had a peak of viremia, and after some days, 28 days, most of them cleared the infection from the blood. But some of them had a rebound. Most often, this rebound is associated, or is said that is associated to escape mutants variants that were able to escape the immune control. Okay? We did some work on that. And for understanding how these escape mutants can happen, what we did was to compare the infection in different types of animals. Okay? These are naive pigs, animals with no previous immunity. And when we examined these animals with no previous immunity, what we did was to put in contact naive animals with infected animals. And by doing what we call the next generation sequencing technology, a new system of sequencing, we examine each and every the variation and in each and every position of the genome. So these points here, and these represent a given nucleotide in the viral genome, were the positions that were transmitted preferentially in the animals, and there with these were the positions that were not transmitted. In other words, variant viruses covering these positions were transmitted better than viruses having these positions here. Okay? If you look at these points are more or less scattered along the genome. Let's see what happens when we try to do the same with vaccinated pigs. The animals that we examined here were vaccinated pigs. They had some level of immunity, and they were put in contact for 21 days with animals that were infected, that were viremic. Can you see that the points that were selected for mutation or for change accumulated in very few points of the genome? Basically, in the NSP2, in the NSP9, that is the replicase of the virus, in the OF2, probably because OF2 harbors one of the neutralizing epitopes of the virus, and in ORF5 and ORF7. So this indicates that probably the animals that have some level of immunity can be infected, and the variants that infect these animals are different than the variants that are selected for transmission in naive pigs. So indicating this role of immunity for selecting these variants, and this is nothing surprising. Immunity is, again, one of the driving forces for evolution of viruses. So some questions about this significance of these findings. The first one, is recombination important for the generation of new PRRS strains? And the short answer is yes. The long answer, yes, but we don't know how important it is. We don't know if this is contributing to 10% of the new strains or 5% or 50%. We don't really know. The only way to know that is to accumulate more data. The second question, how can we gain understanding on the importance of recombination? First of all, by surveillance. We need to collect data from the field. And not just OR5 data, but whole genome data. And with this knowledge, maybe we start having forecasting capabilities. At present, we are unable to forecast anything except saying that we have recombination in the field. The third point, and I think that this is the one that brings you here today, the use of live vaccines and recombination. For this, we have to consider a couple of things before discussing further that. The first one is, why are we using live vaccines? We are using live vaccines because the immunity, if we want to do a primary immunization, the first immunization, priming of the animal, the potential of live vaccines is much better than that of inactivated vaccines. When you start a vaccination protocol for pets with an inactivated vaccines, results are very poor. A different thing is if you use this inactivated vaccine for boostering. That's a completely different thing. But for starting a protocol, you need a live vaccine for producing a substantial immunity for pets. The second thing that we have to consider, how is this recombination produced? For having recombination, we need an animal being co-infected by two different viruses. And here we have a very simple idea that I would like to transmit you. One of the ways in which we can deal with recombination, linking this 3 to 4, is just by sticking to simple things. Vaccines are made for prevention. And prevention means you have to use it before the animal is infected, not when the animal is infected or after the animal is infected. So that means that by using rationally these vaccines, that means by using vaccines in healthy animals, non-viremic animals, you will seriously limit the impact of this recombination. You have to be aware of that. This is a matter of probability. If you vaccinate lots of animals that are infected, sooner or later you will have a recombination. If you stick to the protocol of vaccinating healthy animals, non-viremic animals, and letting the animals to develop immunity for enough time, you are limiting that. So this brings us to the concept of risk efficacy. Can we produce without vaccines for PRRS or for many other diseases? The answer is, well, probably not or very difficult. Can we use these vaccines freely as much as we like? The answer is no. We have to use it on a rational way. And just for the last part of my talk about sequencing, many times people think of sequencing as a diagnostic tool. Indeed, it's not exactly that. It's a complementary tool for diagnosis and monitoring. So the value of sequencing is not produced by the sequence itself, but by the accumulation of sequences in the available databases. I think this is very important. I mean, we need to collect data, to analyze data, and to provide a meaning, to extract a meaning from the data that we are accumulating. What this sequencing can do for us, they can give us a characterization of this strain on a deeper level, not just type 1 or type 2, or just I have a sequence that is a vaccine or something else, but I can have epidemiological information from this sequencing. And the epidemiological information is probably the origin of this strain and the prioritization of measures. That's very simple. The thing is, if in my farm I'm doing things for controlling PRRS and over and over and over and over again, I have the same strain by sequencing, this is telling me that I'm doing something wrong because I'm not controlling anything. And I'm doing something wrong inside the farm. If I'm sequencing and I find in the farm a new strain, this is telling me that I have some biosecurity breach in my farm because I'm letting the virus from the outside world coming into my farm. And this can, maybe it looks to you very simple, but this is really important. You cannot control infection. You cannot control PRRS without a very good biosecurity, internal and external, in the farm. So if you have to do a prioritization of the measures, you need to know where they will come from. How is this sequencing performed? The most classical approach is the Sanger sequencing, that is kind of a PCR reaction in which the nucleotides that we add are labeled with a fluorescence marker, and we take advantage from the fact that these nucleotides are in the dideoxy form, and that means that every time one of these nucleotides is incorporated, the reaction stops. So we can, and you can see in the next slide, we can produce this type of chromatograms in which more or less what we are interpreting is position number 1 is, I'm sorry, but I cannot see the colors from here. It's from some color, this other is from a different color, and so on. And that means I have a T, a C, an A, a G, or whatever. So I can produce the sequencing of that. Normally, most often, we are using OR5 or OR7. And why we are doing that? The origin, the very, very, very origin of using OR5 for sequencing is that OR5 is variable enough to allow us to discriminate different clusters, but also has segments that are conserved enough to produce a meaningful clustering. And I think that this is very easy to understand that. I mean, if we only look at the differences, every one of us is different from the others, but we also share things in common. So that's the idea behind clustering. We have to be able to differentiate, but not that much to have units, And we have to look at the things that are common, but not that much, because if not, everybody is the same. So that's the idea. The important thing here, if you look at the clustering produced by OR5 and OR7, it's not necessarily the same. And if you bring this to one step forward, I know that you cannot read anything from this slide, it's very small, but anyway, the thing here is that they are using, these people, Dormans and co-workers, it's a recent paper from them, if you're using ORF2, OR3, OR5, OR6, OR7, or whatever you're using, clustering of these strains may be different. And if you go back to the picture where we were showing you the recombination, one of the reasons for a different organization of the different segments can be recombination. So we have to move a little bit forward from Sanger sequencing of limited segments to maybe a broader sequencing for having bigger information, for having deeper understanding. One very practical thing, what type of samples for sequencing, and most often you use blood, I understand, for testing the animals, but sometimes you're using oral fluids. And sometimes you test adult animals, and sometimes you test young animals. Let's take a rapid look at that. One thing that we shouldn't do, of course, testing pools of samples. And oral fluids in this context are an aggregate sample. Why? Let's imagine that you have two different strains in your head, and you are pooling the samples. What I would have here? Normally when you do that and you have two strains, you have something like this. Can you see here that you have two different peaks at this position? You have two signals at the same place. Maybe it's blue and red or something like that. I cannot really see that. But the idea is, if we have to produce a sequence must come from an animal. If we try to produce sequences coming from pooling of samples, we are mixing the signals that we have from different animals, and that can create errors in the samples that we have. This one is more clear for you. We have double peaks, double signals in many of the positions that we have here. Is this a sequencing error? Do we have two strains? What do we have in there? That cannot be interpreted just straight from this result. The animals. Remember, sows, short viremia, suckling piglets, long viremia, nursery and fattening pigs in the middle. That's really important because many times you see farms where they bleed sows and tell you, can you test these sows by PCR? Can you sequence the virus? And most often you got nothing. So be careful. One thing is a sow that aborted yesterday, probably is positive, and another thing is just doing monitoring in the farm. That's a completely different thing. Take care with that. One important point are you familiar with this term, CT value? Okay. We are using now real-time PCR, and most often you have a result that says sample positive CT 32, 25, 37. Okay. This value more or less accounts for the amount of viral genome that you have in the sample. Values lower than higher than 30 to 33 probably cannot produce sequences of any value. Be careful with that. Good sequences come from samples 20-something, 30 maybe, but not more than that. And I was explaining a little bit about this whole genome sequencing, and the approach that we take now most frequently is the RNA sequenciation. We are able to sequence right now directly from the sample if the sample has enough virus. And the approach is completely different because in the other system, in the classical system, was kind of a PCR reaction. This is not a PCR reaction. What we are doing is just breaking our RNA, linking this RNA to what we call an adapter. This is the most common way that we do. It's a small piece that we put there, a small piece of nucleotides, and then we start using random nucleotides to analyze the whole sequence. So it's a complete difference. I mean, let's imagine, whole pure genome, Sanger means about 22 to 25 PCRs for doing that. Next generation sequencing means extract the RNA, send the RNA to the lab, and that's all. Completely different approach. Okay, I think I'm on time, so thank you very much.